Genotyping technology has changed an enormous amount in the last 4 or 5 years. In the maybe 10, 15 years ago, DNA marker technologies were quite primitive, and they didn't use methods where you actually obtained the DNA sequence of the individual. But in the last 5 to 10 years, DNA sequencing technology has improved so much, so that actually DNA sequencing is so much cheaper now, and it's actually possible now to directly sequence every individual that you want to look at. Genotyping by sequencing is a new genotyping method which has been developed in the last 5 years, which involves actually using directly the sequence of an individual's DNA as a marker. This was not possible until very recently. In G2P-SOL, we have decided to focus mainly on the use of genotyping by sequencing. This is partly because the cost of sequencing has now made it possible to use genotyping by sequencing in the project, and also genotyping by sequencing offers a level of detail that was not possible with other marker systems. And actually, genotyping by sequencing is a generic method that is particularly suitable for analyzing large numbers of samples of plants. So some of the difficulties of analyzing large numbers of plant samples is that plant species are very diverse, and they also many plants have a lot of compounds in them which actually make the isolation of clean DNA, which can be used for genotyping, quite complex and quite difficult. And so actually one of the key steps in doing genotyping by sequencing, or any kind of genotyping, is actually to get good quality DNA from plant material. And this can be challenging. So this is one challenge. A challenge to doing genotyping in plant material is how to sample from plant populations. Plant populations are incredibly diverse in their genetics, and sometimes it's not just sufficient to sequence a single plant from a population, because it doesn't really represent the entire diversity. So one of the challenges is actually to sample enough plants from a population or a species to actually represent fully the diversity in that population. And this is one of the things we really have to think about in G2P-SOL, about how best to sample individuals.